Okay.
Okay.
Dr. Anurag Jyoti ji, you're most welcome. He's from MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Maharajpura, Gwalior. He will be speaking on advances in waterborne pathogen detection, culture plate to nanotechnology. The stage is
Ma'am,
yours.
should I share my screen?
Yes, please.
Thank you. Ma'am, is this visible?
Yeah, it's visible and clear.
Thank you so much, ma'am. At the outset, I would like to thank organizers for giving me this opportunity to share my research findings. I'll be talking about my research findings related to advances in waterborne pathogen detection from culture plate to nanotechnology. So, myself, Dr. Anurag Jyoti, working as an assistant professor in Amity University, Gwalior, in Department of Biotechnology. So, next few slides, I'm going to kind of cover the recent findings of my research, what we are doing here. So, this includes introduction, rational objectives, work done, and conclusion. Coming to the introduction, as we know that the emerging pathogens causing diseases are global threats, and this holds true not only for the other kind of diseases, it is also true for the waterborne diseases. So, a lot of waterborne, foodborne diseases which are prevalent in environment, they themselves create antibiotic resistance, so that they further complicate the treatment procedures. So, as per the WHO report,
Yeah, रखती जा रही है.
listed among the top 5 death-causing diseases,
Sir, इनका वो कहाँ
causing
गया?
4.3%
Second
deaths.
page.
Again, as per the WHO, the
ज्योति
another disease
श्रीवास्तव
which
जी
is
को, जिसका हम लोग के साइन हो
typhoid.
गए.
So, over 2.16 billion episodes of typhoid occurred
भूल गए यहाँ
worldwide,
से.
which leads to 216,000 deaths, and more than 90% occurs in Asia. So, Asian countries are on the hotspot of these diseases. Major waterborne pathogenic bacteria, basically they have been Salmonella, E. coli, various pathotypes of E. coli. We can see Enterococcus species, Vibrio, Staphylococcus, and many more. So I'll be focusing more towards the above two, Salmonella and E. coli. This is the global
Excuse
scenario.
me.
Yes,
Excuse me. Your slides are not changing.
slides are not moving. Just
Yeah.
right now, is it
Yeah.
moving?
No. Yeah. It's okay now.
Right now, okay, ma'am. So as we can see, I was talking about this major waterborne pathogenic bacteria. So this is the list, and I said that we'll be focusing more towards Salmonella as well as E. coli. This is the global scenario. As you can see that I have taken 3 images. So here you can see that in all the 3 cases, you can see that India has been put on the high risk zone, whether it is traveler's diarrhea, it is typhoid disease, or we have the bloody diarrhea. Okay, so again, I have encircled India, where you can see that India is the hotspot for these diseases. So that's why further they are going to complicate the treatment procedures also. Though we have advanced through a lot of the medical technology, but still we are lagging as far as the rural population is concerned. This is the list of existing methods for the detection of pathogens. We have culture-based established methods. We have for typhoid specifically Vidal method, Typhidot, ELISA-based method, SPR-based method, which is actually the surface plasmon-based method, and then we have RT-PCR method. So these days we know that RT-PCR has become very popular for the detection of SARS-CoV virus also. But at the same time, the problem here is that though the sensitivity is good, as you can see from 10 copies we can target, but it further complicates the detection procedure. It's costly also, and it requires the trained personnel also. So therefore, the rationale of the study is that existing methods, though we have already, so they are labor intensive, time taking. They are challenges of low specificity, low sensitivity, and most important is that they are unable to detect viable but unculturable state of bacteria, which is the actual problem we face generally. So if we do not achieve any kind of result, so we call it there is no pathogen, but that is regarded as the false positive or false negative. So in order to remove this lacuna, we should have the detection system which should work as fast, simple, bedside detection system, quantitative, and that should be highly reproducible. This is the timeline of detection system. As you can see, as for the advancements are concerned, we are culture-based methods, then PCR-based methods, they came into existence. Then real-time PCR became very, very popular. Although these days I mentioned that RT-PCR-based methods have been established as gold standard method, but still due to some lack only, we have to move forward towards the application of nanotechnology. Like in a single essay, we can say yes or no, pathogen is present or no. So it can be reasoned that application of real-time PCR and nanotechnology, this both can together lead towards the development of portable and robust pathogen detection system for on-site detection of pathogens. So these are the objectives. The number one is for my study, we have chosen real-time PCR-based probes for detection and tracking of bacteria in water. And second is the nanoprobes, nanomaterial-based probes for the identification of pathogens. The probes I have used in this study, we have designed and developed these probes. The number one is the molecular beacon probe, which is specific for real-time PCR. Okay, as you can see that it is the nucleic acid which is specific to the target. Once it is attached with the target, you can have the signal, and that signal is being read by the camera which is there, and that will give you the fluorescence, and based upon that you can say yes, there is some target. Gold nanoprobes, they are different from the existing ones in a sense that they are simple one, they are fast, and they are very, very efficient also. So in my research, I have first computed molecular beacon probe. Then for that, further, I have generated the standard curve. For this, I have isolated genomic DNA from Salmonella, prepared the standards of DNA as low as 10^6 to 1 genomic equivalent, means 1 copy of genome, and then I have conducted the real-time PCR. And at the same time, also taken the water sample, drinking water sample, and then isolated multi-genomic DNA and run the assay for the quantification. So this is the result where you can see the designing of molecular beacon probe. I have designed the INVA gene molecular beacon probe, which this gene is signature gene for Salmonella. So you can see the structure of molecular beacon. Here I have modified the FAM, which is the reporter of fluorophore. And here you can see the dapsil, which is the quencher. These are the sequences of molecular beacon probes as well as the primers. And these are the conditions on which we have run the PCR. Coming towards the result, as you can see that for the real-time PCR, we can, in my experiment, I have achieved as low as 1 genomic equivalent, means 1 copy of genome has been detected. Look at the sensitivity. Sensitivity is very high with outstanding PCR efficiency. Okay, so here you can see the PCR efficiency of around 93%, which I have encircled. The detection probability, further we calculated, we thought that what would be the detection probability of detecting 1 copy. So in our experiment, we found that 5 times we run the experiment and we found only 1 time we are able to detect the 1 copy of genome. So for 1 genomic equivalent, our result is detection probability is 20%. For 10 and higher, the detection probability is 100%. Means in all the assays, we have achieved at least 10 copy of the genome. And this is the result based on the PCR. There is no need for running on the gel, but still we have checked it through conventional PCR also, that how much sensitive our experiment is. So here on simple PCR, we can achieve 100 copies, whereas in real-time PCR, I have shown you as low as 10 copies have been achieved with 100% efficiency. So these are some of the real-life samples which we have seen that, see the limit of detection, the contamination level of these pathogens, particularly Salmonella. Out of 4 samples, 2 were positive. Next part of my research is colorimetric detection of DNA portion of your E. coli using gold nanoparticle probes. For this experiment, we have synthesized gold nanoparticle probes in our lab with the collaboration of our collaborator. We have selected the LT1 gene. Again, this is the signature gene of E. coli. So here the strategy is, as you can see, that we have taken 7 probes and we have allowed these probes to target the, the piece of gene. And these are the sequences which we have designed. We are covering the gene sequence of 1.2 kb. So it is nearly 1257 number of bases. And this is as far as strategy and designs are concerned. This is how we are going to functionalize our gold nanoparticles with the oligonucleotide probes to form the gold nanoparticle probe. So it's a simple procedure. Gold nanoparticle, you have to incubate it with thiol modified oligonucleotide, and further through series of experiments, we can finally achieve the functionalized gold nanoparticle probes. We have also checked the stability under salt conditions. So above one shows that it's a gold nanoparticle probe So as good as 2 molar, it can be stabilized under 2 molar sodium chloride. Whereas unmodified gold nanoparticle, the moment you put 0.1 molar NaCl, it is highly unstable and it will just agglomerate. So the probes which have been modified, the gold nanoparticle has been modified into probe, is highly stable as compared to the bare one. Now, to further see whether there is an hybridization, the probes are hitting the target. We have seen through transmission electron micrograph. So here you can see that the good aggregates can be seen. Okay, so look at the gold nanoparticles, they have been aggregated. Okay, in initial. Whereas if you look at the control, I'd like to show it here. NTC is non-template control, and EHEC is the other pathogens DNA. So since there are no mismatch, it's a kind of your non-template target, so there is no hybridization, so therefore you cannot find the particles are clumping. All right. Further, to see whether we can achieve this experiment through spot and read. So since I said that the detection system should be simple, it should be cost effective, bedside. So let us have a look that if we are achieving any sort of color change. So that way we have also spotted our hybridization experiments on the reverse phase TLC plate. So here you can see on first the GNP, that is gold nanoparticle alone, which you can see the slightly pinkish color. The second is the gold nanoparticle probe. Again, you can see there is no change in color since there is no target. Third, fourth, fifth, you can see the hybridization has been done and you can see color change. Third, fourth, or fifth, it is more towards purplish or ash color. Whereas compared to the sixth one is also your no template control. So here again, the color is not going to be changed. So the basic thing is that in 1, 2, and 6, no color change is monitored. Whereas in case of perfect hybridization, third, fourth, and fifth, we observe change in color. So to conclude the study, both parts, the highly sensitive and specific molecular Decon real-time PCR-based method has been established using INVA gene. The second one is the aggregation of gold nanoparticles seen in TIM indicates the hybridization and target of detection of targets. The assay developed is rapid, simple, and specific towards their target, since we have also cross-checked with another pathogen's DNA. And the fourth is that the color change visible to the naked eye in spot and read format has the potential to be developed as a point of care diagnostics. Although a lot of work still needs to be done, we are conducting a lot of practicals. We are also in the process of filing patent of this. So I have not shown that part of my research. So thank you again. And again, I would like to thank organizers again, and very happy to have any questions.